All right, so welcome to our first lecture in our class. This is the chemistry of everyday life. This is the first unit. What we're going to talk about is the chemistry principles and some history of the scientific method. So let's start out this lecture with describing what is chemistry. Okay, so chemistry is the study of matter. So what it is uh, and how to describe it, right? So its composition and its properties, and finally how it changes. So that's chemistry. What it is, how it changes. Okay. Some matter we can see and some matter we can't, right? This pot, for example, is something that you can physically see, is big enough for us to see. Some other matter is so very small that we can't see it. For example, in this picture here, the air, you can't see it, but it is still matter, right? The oxygen molecule is still a matter. Another aspect here I want to point out is that everything we do involves chemistry. So let's take a look at this first picture here. We can see that this pot is made of a substance, a metal, and what about that metal substance that makes it a good container for cooking or for melting or for holding chemical reactions, aka cooking food, right? Or the chemistry in the food itself, like this cheese, right? When you add heat to it, it changes its shape. For example, solid becomes a liquid, um, or even the chemistry of how that cheese was made, right? It was fermented from milk, right? And so going over to this picture here, you can see that okay, how was that milk made, right? It was a biochemical process uh, uh, that converts the things that the cow eat, energy and whatnot, and to making the grass and making the milk, right? Or we can take a look at this picture again, and we see that there is um, clouds here, right? So how does the puddle of water become evaporated and becomes clouds in the sky, and that's a cycle of water, right? So that's all the chemistry that surrounds us. Now, chemistry is just but one branch of science. Let's first talk about what is a science, right? Just a basic definition. So science is a process by which we learn about our world, about the natural universe. And we do that by observing, by testing, and then generating a model that explains what we see, right? So that's what science is. And the thing about science is that it can then, based on these findings, make predictions for a different case based on what we saw in the other scenario. Okay, so that's the importance of science. Now, oftentimes chemistry is coined as the central science, and that's because all of these other science, all these other branches, kind of has a foundation, or their understanding is you have to understand chemistry first in order to understand these other fields. For example, um, biology, right, or medicine or geology, you have to have an understanding of chemistry of the molecules involved in all those systems. And sometimes the study of chemistry is so intertwined with another study that we make a new field. For example, if you study the overlap between chemistry and biology, we call that biochemistry, or the study between chemistry and geology, we call that geochemistry. Now, before all these branches of science are the way they are today, or in particular, before chemistry is what it is today, it took on a different form. It was called alchemy. Okay, but this different form was drastically different. So before 1600s, what is known as alchemy was practiced all over the world in various civilizations, but it was quite different because for two reasons. One is it's more of a mystical, magical approach, and oftentimes it's there to try to manipulate the natural world. For example, like changing metal, like iron, for example, into gold, something more precious, right? So it's very magical aspect to it. And then the second part is often it is secretive. So for example, this list of substances was written in codes that is hard to decipher as seen in this image here, okay? Yeah, and then in the scientific revolution is when we basically moved from alchemy into the world of chemistry, among other branches of science, okay? And so now when we talk about modern chemistry, we can break it down to five categories. The first one is called physical chemistry, where we study rates, energy, structure of reactions of molecules. For example, we can study how fast the ozone breaks down in the atmosphere in order to predict the depletion of ozone in our atmosphere. Right? So that's physical chemistry. The second branch of chemistry is organic chemistry, and this branch mainly studies any molecule that contains carbon and hydrogen. Right? So this can range all the way from oil and fossil fuel to macromolecules, meaning big molecules like DNA and enzymes and stuff like that. Right? So, but when we talk about organic chemistry, most of the time people will think of making drugs, right? Making molecules that will become drugs, or designing ways to make those drugs. Okay. The third branch here is inorganic chemistry. So that's the opposite, right? Inorganic, opposite of organic chemistry. And so the rest of chemistry that doesn't have carbon is studied in inorganic chemistry, but mostly we tend to think about in this category are the metals, right? The rocks and the minerals. In the modern times, inorganic chemistry, one big usage of it is to find catalysts or ways to speed up organic reactions. Or another is in our technology, right? In the chips, in our computers, in our iPhones and whatnot. So that's all inorganic chemistry, precious metals. Another category is the analytical chemistry, and this is mainly focused in separating and identifying unknown samples, right? The analyzing part. And lastly here is biochemistry, which is focused on living systems. So studying how the chemistry is in a cell. Now, biochemistry and organic chemistry together are the primary types of chemistries that it's kind of used in studying medicine, in treatments, in making drugs and stuff. All right. Now let's move on to our another big topic in this lecture here, which is the scientific method. And so the scientific method here says it's basically a system to search for answers to questions. If you have questions, we want to have a systematic way of looking for the answers, or more practically, solutions to problems. So we have a flow chart here of the scientific method. And it doesn't have to be in this order. Scientists can interject at any of these and then fill in the gap. But for the sake of like clarity, we'll start on the top here with observations. Okay, and then we'll go in more detail. But basically, we have here the first things you make observations about the natural world. After you make the observations, you try to explain it with an educated guess called the hypothesis, right? With the hypothesis, then you want to test it out, right? So we want to then do experiments. After you do experiment, you analyze it, your data, and then you ask, is the hypothesis correct? Then you can create some kind of conclusion, and that conclusion is either stated in a law or in a theory, and then of course that theory can also be fine-tuned with further experiments, and then maybe modified or not, right? So that's basically the scientific method, and I'll go into detail each one of these. So making observations, there's two types of observations you can make. One is called qualitative observations, and the other one is called quantitative observations. Qualitative observations describe properties or occurrences in ways that do not rely on numbers, right? So just describing things how they seem without numbers. So for example, the outside air temperature is cooler during the winter session, or during the winter season, right? Or table salt is a crystalline solid, right? You have a solid as table salt. Or crystals or sulfur crystals are yellow. The color is yellow. Now those are just observations of the way things are. You can also qualitative describes how things change. So this last example here is dissolving a penny in dilute nitric acid forms a blue solution and a brown gas. See, you're describing the change and you're describing it qualitatively. On the flip side of this, when you make quantitative observations, you are making measurements. And so what are the most two important things in a measurement? Number and unit, right? Any kind of measurement needs to have a number and a unit. A number makes no sense if it doesn't have a unit. So for example, you can say that baby is one. One what, right? What's the unit? 1 month old, 1 year old, right? 1 week old, right? So we need not only number, but also the unit. Some more examples here. The melting point of a crystalline sulfur is 115.21 Celsius, right? You have the number, 115.21, and then you have the degrees Celsius, the unit. Another example is 35.9 grams of table salt dissolves in 100 grams of water at 20 degrees Celsius. All of those are quantitative observations that you can make. Now, after you make the observations, you then can formulate a hypothesis, which is a tentative explanation for your observations. Now, it doesn't have to be correct all the time, but the key point is that it is testable. So let's take an example. You observed that we experienced alternating periods of light and darkness, and we noted, and we also observed that we see light when the sun is around, and when the sun is gone, we don't see and we don't see light, we see darkness. Okay, so we can have two hypotheses for this observation. The first hypothesis is that the Earth, okay, the sun is staying still, and the Earth rotates on its axis every 24 hours. And every time when you face the Earth in that rotation, you see sun, and when you face away, you see darkness, right? So that's one observation. That's one hypothesis. The second hypothesis is that the sun, the Earth is standing still, the sun is evolving around the Earth, and every time the sun is near that surface of the Earth, you see light. And when it's far away from that surface, you see darkness, right? Both of those seems plausible, right? And they're both testable. So they are good hypotheses. So all you have to do now is to test it. And how do you test it? You have to you have to come up with experiments, right? Which is our third step here is the design and to perform the experiments. You basically conduct to test the validity of your hypothesis. You want to do this in a very systematic way. You want to make these measurements in very controlled conditions, meaning that you only want to change one single variable at a time. Because if you change too many variables, how do you know which one affects the outcome, right? Of course, that's the perfect case, but in real life, oftentimes you can't hold everything the same, but you do the best you can to keep it controlled. And then the next step is to then analyze and interpret your data in order to draw your conclusion. Basically asking, is your hypothesis true? Is it valid? And if it is, if the results are sufficiently reproducible, reproducible here means that someone can follow the exact same experiments and come up with the same results. Then oftentimes scientists will print out or will publish the methods in their experiment for other scientists to try to reproduce their data. Some historical stories here is that many times in history, when another laboratory, another group of scientists will repeat an experiment to see if they get the same data, sometimes they did not. And oftentimes it could just be the materials they were using were different. And sometimes that in itself, using a different material and therefore not making the experiment reproducible, if they are a good scientist, can then see what about that experiment that did not work, and therefore make a new discovery, right? So that's one example of reproducibility. That's a good one. There's another example, and even in modern times, a negative example of reproducibility. When some scientists say they have made something or say they have discovered something, someone tries to reproduce it, it never works out, and it turns out that because that first group of scientists made up their data, right? So if something is not reproducible, it is not dependable. Whenever you have results that are reproducible, you can then maybe summarize it into a law, which is a verbal or mathematical description that describes a phenomenon that allows for general predictions. Remember, the key point about science is that you can make predictions, right? A law simply says what happens. It doesn't say why. A theory is the one that attempts to explain why something happened, why nature behaves the way it does, right? This is not to say that a law is better than a theory or vice versa. There's just different things, right? A law versus a theory. So know the difference. Well, so here's an example. Yeah, example is that evolution is a law, that animals will evolve over time, while evolution by natural selection is a theory. Theory explains that animals evolve the way they do because of natural selection, right? So that's the difference between a law and a theory. All right, so let's take this in action, right? So what is a scientific method look like in your daily life? Let's say you observe that, every time you wake up in the morning, there's trash overturned and junk spread around your yard. So you form hypothesis that some raccoons are messing around with you at night. So how do you test this? Well, maybe you can install a night camera, or you stay up all night to see for yourself. So the results will either say yes, it was raccoons, or no, it wasn't, right? So that's the scientific method in action. Very simplified version of that. Here's another one that's more real life science. So let's say that there's an observation that the sea levels are rising, meaning that the ocean water level is creeping up higher and higher every year. So what is the reason for this? What is the hypothesis for this? And one hypothesis is that there is a rising temperature in the oceans are playing a major role in the loss of Arctic glaciers, melting and therefore adding more water into the ocean, making it go higher. So how would you make experiments for this? It's over some period of time. For example, in this one study, a 5-year period, NASA surveyed Greenland to measure the ocean temperature and the ice thinning measurements, and just the depth of the ice over time. And so the conclusion, after analyzing the data, the conclusion was that they discovered glaciers extend deeper beneath the ocean surface, more so than once thought. So the more glacier is in the water, it makes them more vulnerable to the temperature of the water, which is warming up, right? And so if it's warming up, it's melting, it's losing its mass, and therefore increasing the mass of the water in the ocean. So then from that conclusion, it contributes to this massive body of work that says that there is evidence of climate change that is caused by humans. And indeed, one such change is the rising temperatures of the ocean, causing the rise in sea levels. So now let's move on to describing further the two types of scientific research. The first one is called applied research, and this involves the studying a specific problem. You have a problem in society. The problem often is in industry or the environment, and you try to find a way to solve that. Right? It's very solution oriented. So oftentimes you think about like drug companies, that's applied research, or medical, the hospital studying cancer, that's very applied research. They have a problem they want to solve, and therefore they use a scientific method to find the answer through research. Our classic example from history here is Mr. George Washington Carver and his work with peanuts. He was trying to study alternatives to cotton as a crop because it turns out that cotton was quite damaging to the land. And so he was researching other crops that were more at least neutral or beneficial to the land. And then from that, he made many discoveries of using peanuts and also using of waste materials like recycled oil. So that was the applied field, but it turns out that he, as a side note, he's one of those earlier people who championed this awareness of how interconnected humans are to our environment and how we have to be a steward to it, as he highlights the mutual dependency of the animal, us, vegetable, and mineral kingdoms, how we're all interdependent. So again, that's a little side, but George Washington Carver was indeed an applied researcher. Now, to juxtapose that, to contrast that, we have something called basic research, which involves the search for knowledge for its own sake, right? Instead of having, oh, I have a problem, what's the solution? Look for the solution. It's, I have a question, I have a curiosity about nature, what's the answer? So that's what basic research is. Not to say that it's not important, right? It's not just for fun, although it is a driving force. The coolness, the intrigue of nature drives it, but oftentimes it will lead to someday being applied to solving the problems that presents itself in the future, or people find different applications for it to address a already current problem. So one example here is Gertrude Elion, who studied purines, just molecules, and their role in the cell, right? Just studying this random molecule. But it turns out later on, so that's, again, basic research, but later on that work contributed to understanding of medical treatments and cancer treatments in particular, and which she therefore was awarded the Nobel Prize in. Another prominent example is Douglas Prasher, I think that's how you say his name, who was the first person to study GFP, green fluorescence protein, which is a protein found in these brightly colored jellyfish, which fluoresce or lights up in darkness, right? So he found it, to be cool, and extracted the DNA out of jellyfish in order to isolate this one DNA that codes for the fluorescence protein, the protein that lights up, right? So why do we need to study this jellyfish, right? How is that helping society, humanity in general? Well, it turns out, fast forward a couple decades, that protein was then fused with other proteins. For example, it was fused with a tumor complex. And then so therefore you can visualize the tumor, because once you kind of shine a certain light on it, it will fluoresce, and then you can visualize this tumor in this mice, for example. And so you can see the progression of the tumor growing, or when you apply certain drugs, you see the tumor dying off. So it became a very important tool in medical research later on. So much so that actually Prasher did not win the Nobel Prize for it, but the people after him who studied the protein and applied it from that basic research did win the Nobel Prize. And it turns out it's quite a sad story, and another tangent here. It turns out that when they gave out that award to the other researchers for studying GFP, the Nobel Prize, they found Douglas as a courtesy van driver for a car dealership. It turns out that because he was doing basic research, and maybe because of other unfortunate events, he basically left the lab, left academia, left his research, and was not doing research anymore, right? Was not a scientist anymore, because it ran out of funding. When you don't have money, you can't really do much. But luckily, because he was the first one to find this gene, now we can have a very powerful tool in medical research. Yep, so don't brush off your basic research. All right, so having said that, let's now go kind of cover some basic terminology in chemistry before we kind of go deeper into the semester here. Then this will round out our lecture for today. As we said before, chemistry is a study of matter and its changes. So what is matter? Matter is anything that has mass and therefore also volume, okay? So what is mass? Mass is very similar to the concept of weight, right? So mass is a measure of the amount of matter in an object. So the more matter you have, the more mass you have. Now, weight is very similar it is a measure of the gravitational force on the matter in an object. So basically, it's like weight is how we see mass depending on the on our atmosphere. For example, mass will not change, but your weight will be different on Earth versus on the moon. Okay, so I just want to, oftentimes though, we use these words interchangeably, but do know the difference between mass and weight. Okay, and then let's talk about now some properties. The first one is called physical properties, and these are characteristics of a substance. So how you describe a substance, that's basically its physical properties. So for example, what color is it? How much mass does it have? What is its density? Basically, will it sink or will it float in water? These are all physical properties. Opposite of this is something called chemical properties. And these are characteristics that describe how the matter changes. Okay, so let's clarify this concept with examples. Let's say, like, what's a chemical property of iron? One chemical property is that it rusts. It combines, the iron molecule or the iron atom combines with oxygen to form something else called iron oxide. That's what rusting is. Or let's talk about carbon. It can burn, so that's its chemical property. When it burns, it combines with oxygen to form with carbon dioxide, and also a lot of energy, right? Fire. How about another substance called carbon monoxide? What is its chemical properties? Well, it is toxic, right? And why is it toxic? Well, its chemical property is that it combines with hemoglobin, therefore depriving you of oxygen. Lastly, here we have neon. Its chemical property is that it's inert, meaning it doesn't react with anything, right? So all of these will have a chemical properties, and chemical properties are linked with chemical changes. Happens when your substance become a different substance. For example, iron become iron oxide. So that's a chemical change. I don't know what happened to that slide, but that's when you change in identity, you have a chemical change. Whereas when you only change in your state, that's called a physical change. Your identities stay the same. So for example, you know about water, right? Or ice. When ice melts, it becomes water. It turns out the molecules doesn't change. It's still H2O, right? 2 hydrogen and 1 oxygen in a solid state is solid water, ice. When it melts, it still is 2 hydrogen and 1 oxygen per molecule, per compound. It's just in a liquid state, right? So that is called a physical change when you haven't changed your chemical identity. Whereas again, over here, we said iron becomes iron oxide. Iron becomes iron oxide, right? It becomes two different things when it changes. So that's the difference between a chemical and a physical change. Now, speaking of melting, let's talk about the states of matter. We have here 3 states of matter, really. We have the solid, the liquid, and the gas state. As you can see from the picture here, a solid has a definite shape and volume. So you have this cube. Here, it has its shape. Its shape will not change, and its volume will not change, right? So that's a solid. Whereas a liquid, as you can see here, will have a definite volume, but its shape is not definite. Its shape depends on the container it is in. So for example, if you have, let's say, a gallon of milk in a container, it has the shape of the gallon of the container and has the volume of a gallon. Now, if you pour that milk into a pot, for example, now it has the shape of that pot, but it still has 1 gallon of milk, right? So its volume is the same, but its shape is not. Now, when you move on to the gas, nothing is the same, neither its volume nor its shape. So imagine here you have a gas in a canister. It's compressed in that small volume now, and in that shape of the container too. Now, if you release that gas into the room, it will fill up the whole room, right? So what is the volume of that gas? Is the volume of the room. And what is the shape of the gas? Is the volume, is the shape of that room. So gases just doesn't have shape or volume. It just takes on the shape and the volume of its container. So that's the differences between the 3 states of matter. We can further categorize matter into whether it being a substance, a pure substance, or a mixture. Let's first talk about substances. because you can break down substances into two subcategories. It can either be elemental or it could be compound. An element just basically means it's made up of one single element, whereas a compound means that substance is made up of multiple elements. So, for example, here, this coin, this gold coin, is made of only gold atoms, whereas this glass of water is purely water, but each water molecule is made of, as we see before, two elements, hydrogen and oxygen. Right? So that's the difference between pure elements and pure compounds. On the other hand, you can have a mixture. So you can have a mixture of the elements and a mixture of compounds. But the different mixtures you get, there's two types of underneath this category is homogeneous versus heterogeneous. Homogeneous means all the same throughout. So if you have an equal distribution of different atoms, for example, this brass is made of different elements, but it's equally distributed. Whereas this brass ring with this, let's say, gold ring here, it's not equally distributed, right? There's more gold here, and there's more brass over here. A more easy example between homogeneous and heterogeneous mixture is take a look in this glass of water again. So water is H2O, right? If you add in sugar in there, sugar compound, and you dissolve it, do you have different compounds in there now? Yeah, the sweetened water is water and sugar, right? If you mix it well enough, it's homogeneous. Can you see the sugar from the water? You can, right? If you take a straw and you sip it from the top, sip it from the bottom, it tastes the same to you. It looks the same to you, no matter where. So it's homogeneous. It's equally distributed throughout. So heterogeneous, though, is, for example, if you take that glass of water again and you just add that gold coin in there. Can you see the gold coin from the water? You can, right? So that's why it's heterogeneous. It's different. You can tell the different parts of that mixture from each other. So that's the easiest way to understand homogeneous and heterogeneous. So again, these slides are just gonna reiterate those definitions we just said. Elements are composed of one type of atom. So you go down to the lowest core of any substance, the smallest unit is called the atom, right? The smallest particle of an element. We'll go over these concepts, the atom, obviously, in much more detail in subsequent lectures. For now, let's go over the definition. And elements are represented by chemical symbols. An example we talked about was gold, so that's represented as AU, or the brass has copper in it, it's represented with Cu as a chemical symbol. Compounds are made of two or more elements. We just talked about elements. When there's two or more of them chemically combined, we call that a compound. So some examples of compounds are sodium and chloride can combine to form table salt. H, or hydrogen, and oxygen can combine 2-to-1 to form water. It's not only a compound, but it's a molecule. We'll talk about that in a later lecture too. And if you have, for example, 6 carbons, 12 hydrogens, 6 oxygens combined in a certain way, we call that a sugar molecule or a sugar compound. Okay, so compounds versus elements, they're both pure substances. It's just that compound has different elements in it rather than just one element. And mixture are blends of two or more substances, either elements or pure compounds that are mixed together. And homogeneous means they are uniform in composition. Every part of it has the same pattern, the same makeup. Whereas heterogeneous is not uniform. You can tell that it's not well mixed. It's another way of thinking about that. Okay, I think that is it for today. So that rounds out our first lecture in this course. Just a little introduction to what is chemistry, what is a scientific method, and some kind of introductory views and categorizations of the thing we study in chemistry, which is matter. I will see you next week.